This is Validation Science, Unit 1, Lesson 3, Strengthening of the Regulations. So in this lesson we're going to be covering the GMPs from the 1970s and beyond, what SISPQ is, a little bit about the Barr case and some recent regulatory changes. In 1982, a product called Tylenol, which would be very widespreadly used in the US, was on the marketplace. And what happened was there was a tragedy where Tylenol was tampered with in pharmacies. So somebody broke into pharmacies and put cyanide capsules into the Tylenol bottles. At that time, Tylenol, and no pharmaceutical to be honest, had tamper-proof seals on them. At that time, some of you who might be older may remember that you got your medicines in a brown bottle with a wad of cotton wool or something on the top and the lid on the top from the pharmacy. 7 people died within hours of taking this Tylenol, and it caused a huge investigation in the United States. Johnson & Johnson were the company who manufactured Tylenol, and they obviously investigated an immediate nationwide recall across the whole of the US for every bottle. So it was a huge undertaking, and it was seen as one of the best examples of PR from a pharmaceutical company ever, because they really took it in hand and acted immediately. As a result, Tylenol managed to hold its market share because of the way that they acted so promptly. The investigation showed that the product was tampered with when Tylenol bottles in the pharmacies, they were deliberately contaminated with cyanide because there was no tamper-proof on them. So this came up with the first tamper-proof seals that the industry had seen. And as we talked about before, with the C in GMPs, now because it's current and it's because it's best practice, all companies are expected to implement tamper-proof seals. J&J were the first ones, and soon it became the industry norm. In 1983, the regulations were amended. As you can see, the regulations are very reactive. It's not in any way proactive. Something terrible happens, the regulations get amended. Another terrible thing happens, they tighten them further. Another terrible thing happens, they tighten them again. So in more recent decades, we're trying to get to a more proactive approach to how the regulations are being amended. So in 1983, anyway, the first Anti-Tamper Act came in, and here we had the requirement to put tamper-proof seals on your products. It also made it a crime to interfere in any way with packaged consumer products. And the FDA issued tamper-resistant packaging regulations for all over-the-counter products. And these were incorporated into the GMP regulations, as you can imagine. So in the 1970s and beyond, there was a couple of significant events occurred. So the GMPs, very much as we know them now from the Code of Federal Regulations, came about in the 1970s. And the GLPs, Good Laboratory Practices, in 1979. A lot of the GLPs really applied to the clinical trial labs more so than industry labs. The ECGMPs came into place in 1989. So you'll see whatever happened in the States was pretty much being mirrored over in Europe, as well as the kind of regulations try to stay current with each other. The PIC and the World Health Organizations revised their requirements in 1992. Part 11 electronic records came in 1997. By 1997, a lot of our processes were being automated, a lot of computers were being used to calculate test results, so they needed to put in place a requirement that put some boundaries around how we use computer systems, and that was Part 11. The GMP guides for active ingredients came in 1998, so that was used for active ingredients. In the 1980s, the FDA started to issue guidance documents. So an example of that would be the guidance on process validation that they issued in 2011. Unlike a regulation, the guide is not legally binding, but it reflects what the current agency is thinking and what their expectations are. So in reality, it's expected that you do what's in the guidance, and it would be very ill-advised not to. The characteristics of a product made according to GMPs are often abbreviated into SISPQ, and you'll see this in the regulations a lot. You saw it in our previous lesson when we talked about how the regulations were formed and what they required from drug products. So the S stands for safety. So you expect that a product is free from any unexpected side effects. So through the clinical trials, the company has ascertained what side effects could be expected from their products, and they're classified as minor, major, bearing in mind the significance and the frequency that they occur in. So that's what the safety stands for. The identity is that it's exactly what the label claims, that you can't end up having a mix-up in the plant. You won't find a different product in a different bottle, things like that. The strength is that it delivers the correct dosage and potency. So obviously that is really important, because if you were getting the wrong amount of your product or your active ingredient, an under or an overdose might cause detrimental effects to the patients, depending on what the product is working on. So strength is really important. Drugs obviously need to be pure. So as I said before, there are 3 different types of contaminations you can have. You can have the physical, so flies, dirt, dust, pieces of metal shavings, anything like that, that's a physical contaminant. You can have your biological contaminants, so your microorganisms, your molds, anything like that. And then you can have your chemical contamination, so other detergents, other products, leftovers from waste streams, things like that, your chemical contamination. Quality, the overriding thing, that it meets all the standards and it can be made consistently. And consistently is a word that comes up in the regulations a lot. That's kind of the cornerstone of GMPs, that you can consistently show that if you do things the way you're supposed to, that your product remains consistent all the time. So you're guaranteeing your quality. The cost of medicines. By the early 80s, there was mounting public pressure to accelerate consumer access to generic drugs. So obviously they're more widely available and they're a lot cheaper also. And up to now, the originator's patent had to be expired before an application could be made by a generics company. So in 1984, the Hatch-Waxman Act was brought into place, and it was the start of generics in the US. They were allowed to put in a special abbreviated application for licensing generics because the product had been on the market for such a long time. You wouldn't have had the same volume of testing to do. You weren't issuing a new product, just a new formulation. There was a 180-day exclusivity for the first to file a generic application. So if you got in there first, you got 180 days to get your product out onto the marketplace before the next generic could come along. If the case for patent infringement was upheld, then the person that took the liability ended up having to pay the litigation costs as well. There was a 30-month hold in favour of the branded companies if infringement was filed. So there was some bit of protection for the branded company. In the 1990s, generic drug bribery and fraud was being addressed. So Par Pharmaceuticals, they tested the brand name product and they submitted these results in their amended new drug application. The employees falsified sorts of analysis, and everybody that was involved in that was debarred from working. So the FDA can debar you from working in the industry, which means you are no longer legally allowed to work in the pharmaceutical industry. That wouldn't be something that would regularly occur. It would be only in very serious cases, and generally it would only be the top levels of the company. But in this case, because it was so serious, they actually included everybody who was working there, and not just the executives. In 1992, the Generic Drug Enforcement Act came into place, and here again we can see that they put in options for debarment people from the industry and other penalties for falsifying a new drug application. So that would be an amended new drug application. That would be when you're filing to the FDA for your new products. The message was sent out to the industry about the consequences. So things that were happening that were being completely frowned upon by the FDA, not recording thoroughly and accurately. We always have talked about how important documentation is in a pharmaceutical environment. Making false entry, so you're actually signing legal documents when you're signing a batch record or a lab testing record. They're considered to be legal documents. So if you put a false entry on it, it's considered to be making fraudulent information on a legal document. Falsifying dates, again, to make something appear like it happened sooner or later than it actually did. Again, a major no-no in the pharmaceutical industry. Backdating, same thing, making things look like they happened in the past. If you forgot to do something or you didn't do something when you were supposed to, changing the date to make it look like you had. Again, legal documents. Signing for somebody else. So in a different industry, that might be an acceptable practice, but in a pharmaceutical, it isn't. It's a legal document. You put your name to it. You only sign the work that you do, and this would be a major no-no to sign for somebody else, either electronically or on paper. Making up data, I mean, obviously you can't do that. Signing for something that you didn't do, so signing to say you did a calibration when you didn't do a calibration, or you did a check when you didn't do a check, again, legal documents, and it's fraudulent. In the 1980s, there was widespread disregard of the GMPs. Things had lapsed quite significantly. And in 1992, the Barr case came about. So the FDA took a landmark case against a company called Barr Laboratories. They found serious anomalies and infringements when they audited their site. They found that records had been misplaced, that test data was recorded on scraps of paper and not into the legitimate, proper worksheet, batch record, laboratory notebook as it should have been. There was failure to control manufacturing steps. They found that products that were released didn't meet their specifications. They also found that when they were doing investigations of products that had failed some testing, they were completely inadequate. And they weren't validating their test methods and manufacturing processes, including their cleaning processes. So they found huge issues in the Barr labs. This really was a huge landmark case for the whole industry, and there was major repercussions across the whole industry, and a lot of companies looked very hard on their laboratories and really stringently audited what was going on and what were the practices going on in their labs, and it tightened up how laboratories were being run at that time. The results of the Barr case was that the FDA can now force a company to recall products. Prior to that, you could do it voluntarily, or they could get the marshals to take them off the marketplace for you, which meant that the state had to bear the cost of the recall. Now the company was able to be forced into doing it themselves. It also put systems in place to correct deficiencies that were found, so there had to be a follow-up and they had to correct issues. They also developed an acceptable out-of-specification procedure. So what do you do in a laboratory if some of your test results are out of specification? How do you investigate it? How do you hold your product? What are the requirements you would need to make it okay to release that product after your investigation is completed? So as I said, this had a ripple effect to all companies, and all companies at the time were affected, and there was a major microscope looking down the lens of laboratory practices at the time. Shortly afterwards, the FDA issued a guide on how to handle out of specification results, and this was really useful because it made in concrete what their expectations were, so laboratories were able to update their procedures to make sure that they were compliant with what the FDA were expecting, and as a consequence, the regulatory bodies in Europe also. Some more recent changes, and we have harmonisation with other international agencies. We're going to be talking about the regulators in some of our later lessons, but just to give you an idea, some of the more recent changes that have come about, we've got ICH, which stands for the International Conference of Harmonisation. They've got some quality procedures. Q8, Q9 and Q10 have been game changers for the pharmaceutical industry. We're going from a place where all of our requirements in how we manufacture things were to the letter of the law. Whatever you had put in your filing for your product manufacturing had to be followed to the letter of the law, regardless of whether there was logic in it or science in it. Once it was filed, that was it, and it took a long time to make amendments to it, and it was very costly. The new approach is more scientific and more logic based. So they're looking at trying to create a space where you are very aware of what your product parameters are, and you know what the impacts on your quality are going to be, and you have a lot more knowledge about how your process works. So if you want to make little tweaks that are within the boundaries of that there's ways and means to do it now where there wouldn't have been in the past. So ICH Q9 is around applying a risk-based approach, so making a decision based on science, logic, and the impact on the product, as opposed to just what was mandated verbatim by the regulations. ICH Q8 is looking at quality by design, how you file your product so that you've got a little bit of design space there, that you have quarantined out your critical parameters, and within the scope of what you're allowed to do, you can make tweaks under certain regulated procedures, obviously. And the ICH Q10 is the same. So again, looking at being more logic based, looking at being more interactive between how products are designed and how the scale up of manufacturing happens and what happens in the real world in commercial manufacturing, getting more knowledge base around your product and your processes. So now we're back to you. So if you could just take a couple of minutes here just to check your understanding of the slides we've just gone through. So number one, why do we have GMPs? What were the critical events that helped the GMPs evolve? What does SISPQ stand for? What is the difference between a regulation and a guidance? So thank you very much for your time, and we'll see you again for the next lesson.